Hi dear students, I am Dr. Mashur. Welcome to my class. Today I would like to discuss TI plasmid of Agrobacterium tumefaciens. Okay, tumor inducing plasmid of Agrobacterium tumefaciens. Okay. This TI plasmid which is present in the Agrobacterium tumefaciens. Okay, this TI plasmid which is present in the Agrobacterium tumefaciens. Okay, and the agrobacterium tumefaciens is also known as nature's smallest genetic engineer. Okay, nature's smallest genetic engineer. Okay, this bacteria, actually the bacteria is a prokaryote, and that will transfer DNA into plants. Okay, that will transfer DNA into the plants. Okay, that means gene transfer will occur from a prokaryote to eukaryote. Okay, this Agrobacterium tumefaciens is a soil bacteria that is known as nature's smallest genetic engineer. Okay, that can able to transfer DNA, part of its plasmid DNA, into the plants. Okay, and this soil bacteria, okay, this soil bacteria, Agrobacterium tumefaciens, that cause crown gall disease in many species of dicotyledonous plants. Okay, this soil bacteria, Agrobacterium tumefaciens, that cause crown gall disease in many species of dicotyledonous plants. Then how we can see how this crown gall disease is formed. Okay, when the bacteria uh, invaded the wound on the stem of this dicotyledonous plants. Okay, when the bacteria in Agrobacterium tumefaciens invade the wound of this dicotyledonous plant stems. Okay, here you can see Agrobacterium is this which is present in the soil. Okay, Agrobacterium is present in the soil, and here a wound is formed in the stem of this plant, and this bacteria will be invade this plant cell. Okay, bacteria will be invade this plant cell, and that will form a cancerous growth in the crown part. Okay, this is the crown part of this plant, and the bacteria form a crown part. The crown means a cancerous proliferation. Okay, sorry, crown means gall means a cancerous proliferation. Okay, near to this crown, a cancerous proliferation gall is formed, and that is known as a crown gall. Okay, this is the region of crown, and there's a wall is formed due to the infection of Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens is a naturally occurring bacteria, and that is also known as nature's smallest genetic engineer, and with that can able to transfer uh, part of the plasmid DNA into the that host plant cell. Okay, that means prokaryote to eukaryotic gene transfer can occur in this case of Agrobacterium tumefaciens, and this soil bacteria will cause a disease called crown gall. This Agrobacterium tumefaciens called a disease called crown gall disease in dicotyledonous plants. And then how this crown gall disease is formed? Means how this crown gall disease is formed means here you can see this is a healthy plant, and in the soil you can see agrobacteria is the present, but here you can see a wound is formed in this stem of this plant, and this bacteria invade this wound, and that will multiply in this plant and form a gall. Okay, and form a cancerous proliferation called the gall here near the crown. That's why here you can see, that's why this is called a crown gall disease. Okay, it's called as a crown gall disease. And now we can see how this crown gall is formed. Okay, how this crown gall is formed. There you can see the importance of TI plasmid. Okay, told that this agrobacterium is this soil bacteria that will cause crown gall disease in dicotyledonous plant. Now we can see how this crown gall disease is formed by this agrobacterium. Okay, how it's formed. Okay, the ability to cause crown gall disease in plants is because of the presence of a plasmid. Okay, the ability to cause crown gall disease by the agrobacterium tumefaciens is the presence of a plasmid called tumor inducing plasmid. That means TI plasmid. Okay, TI means tumor inducing plasmid within the bacterial cell. Okay, the crown gall disease is formed in the dicotyledonous plants by the infection of Agrobacterium tumefaciens is the presence of TI plasmid in the bacterial cell. Okay, presence of TI plasmid in the bacterial cell that leads the production of that leads the formation of crown gall in the Agrobacterium infected dicotyledonous plants. Okay, and this this large plasmid, this large TI plasmid having a size of 200 kb. Okay, this large TI plasmid having a size of 200 kb size, and it's carry now numerous genes involved in the infective process. Okay, and this 200 kb plasmid contain, you can see this this plasmid contain numerous genes which help for infection process of plants. Okay, infection process of plants. And one of the remarkable feature of this TI plasmid, okay, the one of the remarkable feature of this TI plasmid is after infection of this Agrobacterium tumefaciens, this Agrobacterium tumefaciens will transfer a part of this TI plasmid, okay, a part of this TI plasmid into the plant chromosomal DNA. Okay, a part of this TI plasmid is in the will be integrated into the plant chromosomal DNA, and this DNA. That's okay. This DNA will be this one part of the TI plasmid that will be um, transfer into the plant chromosomal DNA, and that segment of this TI plasmid is called a tDNA. Okay, tDNA. Here you can see tDNA. Okay, this part of this TI plasmid, this part of this TI plasmid will be transferred into the host chromosome, and hence this part is known as a tDNA region. Okay, tDNA, transfer DNA region. This is the transfer DNA, and this part will be transferred into the host chromosome, and this having a size of 15 to 30 kb size. Okay, this having a size of 15 to 30 kb size. Okay, this tDNA portion having a 15 to 30 kb size. Okay, only this part will be transferred into the host chromosome, and this tDNA portion contain 8 or so genes on 8 or more genes. Okay, normally it's having only 8 genes. Okay, here some are not marked here, and uh, this tDNA contains some genes, and these genes are responsible for cancerous properties of the transformer cell. Okay, these genes are responsible for the cancerous properties of the transformed cell. Okay, that means the plant cell. Okay, this transform. Okay, here you can see one plant hormone auxin is the cytokinin is the. Okay, auxin and cytokinin is the for helping for the cell proliferation. Okay, cytokinin help is the plant growth hormones. Okay, when this auxin and cytokinin will enter into the normal plant cell, then that plant cell will increase the production of auxin and cytokinin, and that's why the plant cell multiplication will be increased, and that's why it's cause a cancerous proliferation. Okay, and this TNA portion is responsible, the genes present in this TNA portion is responsible for the cancerous proliferation in the transformed cell. That means, for example, auxin cytokinin is a plant growth hormone. If this auxin and cytokinin gene will enter into the normal plant cell, then cell production of auxin cytokine will be increased. That leads the production of cancerous cells in the transformed cell. And the gene present in some genes, one gene present in the tDNA region that will synthesize a protein called opane. Okay, called a unusual compound. Okay, produce an unusual compound opane. Okay, the tDNA portion also produce an unusual compound called opane, and this opane gene will synthesize opane, and this opane will used by the bacteria as a nutrient. Okay, when this agrobacteria infect the plant cell, that time tDNA gene will be transferred into the host cell, and in that time, that opane gene produces an opane, molecule opane, and that opane that we use as a bacteria as a nutrient. Actually, this opane will place the TA plasmid tDNA inside the plant cell, and this opane will be used by the bacteria. And then this TI plasmid also contain left border sequence and right border sequence. Okay, left border and right border sequence. Okay, we can see again that the ability to form the crown gall disease is due to the presence of TI plasmid in the Agrobacterium tumefaciens. Okay, the ability of causing a crown gall disease in plants, that is due to the TI plasmid. Okay, and this TI plasmid having a size of 200 kb, and that's carry numerous genes. Okay, that's carry numerous genes. And one of the remarkable feature of this TI plasmid is that contain a portion, that having a portion of called tDNA, that will transfer into the host chromosome, that means plant chromosome, when this agrobacterium infect this plant cell. Okay, that means when bacteria infect the plant cell, that time a portion of this TI plasmid will be transferred into the host chromosome, and that transferring part of DNA is called as a tDNA region. Okay, transfer DNA region or tDNA region, that having a size of 15 to 30 kb size. Okay, that having a size of 15 to 13 kb of genes, and this tDNA region contain a number of 8 or so genes. Okay, this tDNA region contains some genes, and this genes present in the tDNA region responsible for the cancerous properties of the transformed cells. Okay, this tDNA region is responsible for the cancerous properties of the transformed cell. Here you can see cytokinin plant hormone gene is present in the tDNA region, and the auxin gene also present in the tDNA region. When this tDNA region in the plant host chromosome, that time the plant cell production of auxin and cytokinin will be increased. Okay, auxin, cytokinin. Okay, when this tDNA region is enter the host chromosome, that time that plant cell auxin and cytokinin production will be increased. We know that auxin and cytokinin will help for cell proliferation. Okay, cell proliferation, this is the growth hormones. Okay, when the production of auxin and cytokinin will increase the plant cell, that time that plant cell go for undergo for cancerous proliferation. Okay, and another one unusual compound which is synthesized, which is gene is present in the tDNA, as is opane. Okay, this opane will be used as bacteria as a nutrient for the nitrogen source. Okay, for the nitrogen source. Okay, this bacteria will, this opane will be produced by the transformed cells, and this opane will be used by the bacteria as a nutrient. And this tDNA region also contain left border and right border sequence. Left border and right border sequence. The metabolism of opane is a central feature of crown gall disease. Okay, the metabolism of opane, opane is a central feature of crown gall disease, and opane synthesis is a property of a cell that is colonized with agrobacterium tumefaciens. That means if if a cell is colonized with agrobacterium tumefaciens, that time the cell will produce opane. And there are different type of opane is there, and the type of opane produced by plant cell is determined by the bacterial strain, not the plant cell. Okay, there are different types of opane is there, and the opane produced by the plant cell is determined by, okay, the type of opane produced by plant cell is determined by bacterial strain, not by plant cell. Okay, that means in general, the bacteria induces the synthesis of opane that it can catabolize and use its sole carbon and nitrogen source. Okay, in general, if a bacterium induces the synthesis of opane, that it can catabolize and use this opane as a sole carbon and nitrogen source. Okay, and if that means if a bacterium produce uh, octopane, okay, and induce tumor, that bacteria can able to catabolize and use thus octopane. Okay, that bacteria can uh, use octopane as carbon and nitrogen source. And if a bacteria produce napalm induced tumors, that bacteria catabolize and use napalm as a carbon and nitrogen source. Okay, thus if there are different type of pain is there. Okay, and the type of pain produced by the cell, a plant cell, that is determined by the strain of bacteria. And if a bacteria produce an opane, that bacteria will use that opane as a sole carbon and nitrogen source. Okay, that means if a bacteria produce octapane. Okay, if a bacteria causes synthesis of octapane in a plant cell, and that bacteria will use that octapane as a sole carbon and nitrogen source. And if a bacterial uh, produce a cause production of napalm opane in the plant cell, and that bacteria will catabolize and use this napalm as a sole carbon and nitrogen source. Okay, this is about the opane. Okay, and in TI plasmid, okay, in TI plasmid, okay, we discussed about tDNA portion. Now we can see what are the remaining part of this TI plasmid. Here you can see one gene, virulent gene. Okay, here you can see one gene, virulent genes, and this gene is responsible for the transfer of this tDNA portion to the plant cell. Okay, this gene VIR gene, virulent genes, that is responsible for transfer of tDNA into the plant cell. Okay, this virulent gene is responsible for the transfer of tDNA into the host cell. And another one gene here is the gene for opane catabolism. Okay, gene for opane catabolism. Okay, in the plant cell, when this tDNA portion is transferred into the host cell, that time that transformed plant cell will produce opane. Okay, opane, and the bacteria will use uh, this opane using this gene. Bacteria will use this opane as a carbon and nitrogen source using this opane catabolism gene. And here you can see two border sequence, left border and right border sequence. Here you can see two DNA part, that is known as left border and a right border sequence. Okay, this right border is essential. Okay, if this right border is essential to transfer foreign DNA into the host cell. Okay, this right border is essential to transfer foreign DNA into the host cell. Okay, if any mutation is occur in the right border sequence, that means this tDNA cannot able to transfer into the host cell. Okay, this tDNA cannot able to transfer into the host cell. Okay, and the left border is non-essential for the transfer of tDNA into the host cell. And this is the TI plasmid. I hope you understood this class. Thank you for listening.